In vivo experimentation, most commonly known as animal testing, is an investigation method in which a live animal is forced to undergo processes that will develop biomedical solutions to human diseases. Through the years, animal testing has promoted enormous development in medicines, treatments, and cosmetics. Thereby, it is responsible for the creation of vaccines and treatments that have saved billions of lives, becoming an important procedure to achieve medical discoveries and innovation. Although these advances have revolutionized medicine, the innumerable consequences that animal experimentation brings have been left aside due to the accessibility that this type of experimentation provides. In vivo experimentation undergo living animals to procedures that are painful, damaging, and cause long-term diseases. This includes dropping chemicals in their eyes, forcing them to inhale or ingest toxic substances, deafness, blind, burned, stumbled, and infected with viruses that are considered cruel actions in common sense. Despite the fact that a process has been initiated to regulate animal testing, such as the Animal Welfare Regulation, they have focused only on a little amount of the species used in this test, leaving aside non-vertebrates, mice, and rats that represent the 93% of the subject animals. Additionally, a high number of research results from animal testing are flawed. Evidence has proven that 41% of publications based on animal research do not explain or include important information about experimental methods, invalidating the obtained results and wasting not only the investigation but the animal life used. However, regardless of the constant evolution that animal experimentation allows, it does not always lead to favorable and applicable results in humans. In this sense, it is considered the possibility that trials that pass in vivo tests will not work in humans. This is the case of research for new procedures to treat and cure cancer. Statistics from the American Journal of Translation Research in 2014 demonstrated that on average only 8% of the trials that achieve in vivo tests are successfully adapted to human models. But the question is, what do we do now? Use people instead of animals? Well, the answer is yes, but not in all cases. One of the alternatives for animal testing is, in fact, human trials. A great example to this is the creation of the COVID-19 vaccine developed by Moderna. The first clinical trial started with a small group of volunteers to test if the vaccine was safe for human use and to determine its efficacy in later studies. Another alternative is in vitro testing, which uses laboratory devices to verify procedures' efficiency in cells and tissues. As an example, in vitro has demonstrated that sheets of skin can be replicated in tubes and be more useful than in vivo in chemical testing in some cosmetics for skin. Finally, a most recent alternative is 3D printers, especially tissue bioprinting. This innovative technology can be used to replicate the 3D structure of a human tissue, which provides a lot more information about a drug's effect than just using in vivo tests with organisms with similarity with the human body. As some scientists agree, humans are not 70-kilogram rats. That is why animals are not accurate models to predict the human response. As it was explained, alternatives for animal testing exist, are in constant development and exhibit the possibility of reducing animal testing and in the future making in vivo tests unnecessary. What is clear is that the debate is in favor of science development for human health, not for cruel animal test procedures.